Open it back up. Let's see.
Okay. Are we back on?
Yes, with
Should I try sharing again?
not yet.
Why don't I put my slides in a file and send them to you just in case?
Okay, that sounds great. Oh, here we go.
Think you have it?
I have a different settings area that I think. Here we go. Oh, I think I've got it. There we go.
Yeah. Okay. Allow to share window.
Perfect. They moved it on me. That was so mean of them. Some update.
Okay, I've got to get my slides up again now.
Okay.
What's happening?
Yay. Melissa, is Cody on our email list? Let me check, because I was just thinking, I don't have his phone number, but he did come to one right before Christmas break.
I'll get rid of this thing off my laptop now. Gee. Okay.
Do you know Cody's email? I've got the list pulled up here.
I can find it.
Okay.
Yeah.
You're sharing screen. Okay. Still trying to find my slides. They're up on my
Oh, diesel fuel.
there's another thing on my, hang on a sec.
You're good. We have a little bit of time.
Yeah.
Can you go back to his email real quick? Thank you. Oh, there's.
Okay. Now pull my slides up again.
Hey, Caitlin.
Hello, Happy New Year.
Happy New Year.
Happy New Year.
You still can't see my slides, can you?
No.
Share this window. So it's still not up?
No, unfortunately. So we can see your screen right now, but we can't see slides. But if you are able to move your window to where the slides are, then we should be able to see that.
I don't know what the heck this matter is. I'm going to send you a copy of them.
Okay.
All right.
Hi, Laurence.
Hi, Laurence.
Hello.
Hi. I don't think you can hear us.
Yeah, I can hear you.
Hi.
Come on. Come on.
We've stayed up really late for you, Laurence.
For some reason I'm having a problem. Okay. Okay. I've just sent you a copy of my slides, Sophia.
Awesome. Let me pull that up.
I'm not using all the slides that are in this file, so don't worry about that when
Okay.
you get them.
Okay.
Have they come through?
Yes, I'm downloading them right now.
Okay, good.
Right.
Ah, good, you got it. Okay. So you tell me when you want me to start and I'll go ahead.
Okay, I think if we give it maybe 5 more minutes and see if anyone
Yeah,
else
that's
rolls
fine.
in. We
Yeah.
just escaped someone's dissertation defense, and Melissa and I were the first ones to leave, so we'll see.
So I'm making this a little different than my usual type of seminar. I'm not giving much data at all. I'm really making it part professional, part personal, and really talking about my career starting out as a graduate student and how it developed into from a very chemical area to a much more translational area. So I'm not going to put a lot of data in. You're going to have to believe me when I say we have data that supports this.
Okay, well, I think I'll trust the FDA on this, so not an issue. But I think that's great. You know, a big part of this is that we're a self-organized group of graduate students and some postdocs, and we're trying to figure out what to do next. And so hearing about the pathway that someone who has already done it took,
Yeah,
very helpful. So that sounds
yeah.
great.
But I thought that would be more useful for this group rather than giving a hard scientific talk, and you can see, hopefully, what happened to me from my undergraduate days at Bath University, starting in 1964, right up to what happened in 2026 now, I guess.
Oh, that sounds great.
Yeah.
Yeah, I think you and I were emailing about this before, but we have some commonalities in diving into new places and
Yeah, that's right.
yeah. But, okay, so I am not sure if we're going to have more people who roll in as we go, but I think right now would be a reasonable time to start. And
Okay. All
um,
right.
if you tell me to move to the next screen, I can happily do it.
Okay. All
And
right.
yeah, we're very excited to hear from you about your journey.
All right. Well, thank you very much for the invitation and also for letting my relatives in from Stratford upon Avon in England who are there, I think. So what I'm going to do in this presentation is talk about my own personal career, primarily the professional part of it, but I'm going to touch on some personal points as I go through and how they affected the journey, I guess, from a very chemical background to one that has become quite translational and actually bringing drugs all the way through into patients, and now having two FDA approved drugs that we contributed to in terms of development as well as finding out how they work. So if I can go to the next slide, please. All right, so this gives you a basic, I guess, outline of what I'm going to talk about. And I'm going to talk about how I started out on biosynthesis of antibiotics back in 1971 as a new faculty member at the University of Maryland, and how I then moved into the mechanism of action of streptomyces antibiotics, and then really worked in this area for about 30 years. And then in the 1990s, I moved from looking at Watson and Crick DNA to something called G quadruplex DNA. And I'll explain what a G quadruplex is when I'm about halfway through the lecture. And then how we went back to look at some drugs that we had worked on in the early 2000s and were able to show that these were actually quadruplex targeted drugs. And these are actually approved for a number, FDA approved for a number of different cancers, including melanoma, as well as small cell lung cancer and soft tissue carcinomas. And then I'll end up by just giving you a few words about lessons that I learned during this journey. Next slide, please. Okay, so I'm going to start out when I went to Bath University in 1964. And this is a picture of me with my parents back, and I lived in Birmingham at that time, but I went to Bath University. So next slide, please. And this is me as a graduate student. And I'm the one, I guess I can't point, but I'm the fourth one over from the left. And that's right, with the tie on. And I'm next to my advisor, who was called Dr. Heinz Floss. He actually, unfortunately, died a couple of years ago. But there are some people in this picture, even though this was taken, I guess, 60 years or so ago, the person standing next to me on my right-hand side is Marin Speedy, who became the Dean of the College of Pharmacy in Minnesota. And this guy here is on the faculty in the College of Pharmacy in Minnesota. And the guy, two from my right, became a vet. And my two supervisors, my main supervisor was Heinz Floss, and then this was Ulf Horneman, and he became a faculty member at the University of Wisconsin. So next slide, please. So the way I started my career was really following up my graduate work, and I was trained to look at the biosynthesis of Streptomyces antibiotics. And the way I started is there was no internet, of course, back in the 1960s or early 70s. So unlike faculty recruiting now, it was very different back in the 60s and 70s. I interviewed at the University of Maryland. I gave my seminar at 11 o'clock. I went to lunch with the dean at noon, and they offered me the job at lunchtime, which I accepted. And I went back to my postdoc position. And then a few months later, I moved from Vancouver in British Columbia, where I was postdocing in the chemistry department, to the University of Maryland, really with little idea of what I was going to do as a faculty member in terms of research project. So I spent the first few days in the University of Maryland Library on Lombard Street in Baltimore. And after 3 days, I really hadn't found anything I wanted. I was looking for a molecule that I could do the biosynthesis on, but I also was interested in compounds that interacted with DNA, because I had taken just one course in molecular biology as a graduate student, and that had intrigued my interest in DNA as a target. So as I was leaving the library on my third day, someone had left a journal open on the desk, and I looked at it as I passed it, and it had the structure of a compound called anthromycin. And that's how I found my first drug to work on, just by glancing at a open journal at the University of Maryland. So can I have the next slide, please? So this drug anthromycin is shown on the left-hand side, and the biosynthetic precursors were tryptophan, tyrosine, and methionine. And we worked out how this drug was put together by Streptomyces, and we published this in JACS in 1975. And this was my first independent publication, and I actually got a symposium invitation out of this paper that I published, and I went to Hawaii to give a talk. So that was my first presentation, and that was in about 1975, 76. And then, can you go to the next slide? And my father had pancreatic cancer, and he unfortunately died of this in 1975. And I flew back. By that time, I had moved to the University of Kentucky. Go back one slide, please. I'd moved to the University of Kentucky, and I flew back and was there really the day before he died. He was in the Bath Infirmary Hospital. And at the end, as I was leaving his room, he sat up in bed, which he really struggled to do. And he said, "Laurence, this is a terrible disease. You need to do something about it." And that's why I got into the mechanism of action of anti-tumor agents. So can you go back one more slide, please? And on the right-hand side of this slide is the anthromycin, and that's this compound. And we showed that it bound to DNA, and we published this in Nature in 1979. And so this was our first endeavor into the mechanism of action of drugs that bound to DNA. And that's where I stayed for the rest of my career, looking at how drugs that interacted with DNA, how they interacted with DNA, and the biological consequences, and then trying to understand how we could get selectivity for cancer cells. So next slide, please. And the next one, please. So this is my research group at the University of Kentucky in 1979. I'm the guy with the red turtleneck on, standing next to a CPK model that I put together that had anthromycin bound to it. And the guy on my right is a guy called Vince Reynolds, and he went from my lab to the University of Wisconsin and then to Eli Lilly and in toxicology. And I saw him actually just a couple of weeks ago. He was at a toxicology meeting in Phoenix, and he dropped into Tucson. And he retired from Lilly, I guess, a couple of years ago. And he was head of the toxicology program there. And the guy next to him on his right is Stefan Rockham. And he's now still a professor at the Hebrew University in Jerusalem. And the other people I know where some of them went, others I don't. The woman on my right-hand side is Ruby Petrusek, who was the postdoc that published the paper with me on the structure of anthromycin. Next slide, please. So now, how to relate the mechanism of action of this drug that I worked on back in the 70s and 80s to a compound called ET743. ET743 is, and I will refer to it also with its other name, which is tribectedin. And this compound is actually derived from a sea urchin. And a pharmaceutical company in Madrid, Spain, discovered this compound in the 1990s, and I recognized that it had a structural similarity to anthromycin, and I got in touch with a company called PharmaMar, and I worked on this drug with them to determine the mechanism of action. So next slide, please. So in the top left-hand corner is this drug, trabectedin or ET743. And then you see anthromycin is down on the bottom right-hand side. And they both have in common at least two features. In this dotted line is the carbinolamine, which is the DNA reactive part of the molecule. And that is also found in trabectedin. And in the green part of the structure, you'll see again this carbinolamine is encircled. And this is the common structural feature. And all these four drugs interact in the minor groove of DNA. They all have right-handed twists. But there's a difference between the trabectedin and the other three drugs, whereas saffromycin, anthromycin, and naphtridamycin, they all alkylate DNA, that is, they covalently bond, and they irreversibly covalently bond. So once they find their binding site, which is a 3-base sequence, that's where they stay. What is different about trabectedin, it's actually reversible. It's covalent, but it wanders up and down the DNA until it finds its thermodynamically stable site. So that's the critical difference in terms of chemistry between trabectedin or ET743 and the other three drugs. And from a pharmacological sense, trabectedin is far more potent. It's about 1,000 times more potent. It's nanomolar versus micromolar potency, and it's also far more selective. So this is the one that's approved for a number of tumors, and we worked on it as a duplex interactive drug initially. So next slide, please. So this is the structure of trabectedin on the left-hand part of the slide. And this is a very similar drug, but this drug is called saframycin. It's a streptomyces antibiotic or anti-tumor antibiotic. And this is made by a sea urchin. And the critical difference between the two drugs is that the sea urchin actually depends upon this drug for growth. If you knock out the ability of the sea urchin to actually make this compound, it can no longer survive. So there's something very special about this compound. And structurally, the biggest difference is that it has this C subunit, which is at the lower part in a, I guess, it's an orange color. And that doesn't exist in the other compounds. And if you look at the right-hand side of the slide, you see a structure of this compound bound to duplex DNA. And this C ring is external from the helix. And in order for it to actually covalently bond, it has to open up the minor groove more than saframycin. And this turns out to be very critical. So this trabectedin widens the minor groove and it bends the DNA into the major groove. And that is the reason it's reversible. Whereas in the case of saframycin and the other compounds like anthromycin, they covalently bond to DNA and they don't wander up and down. As soon as they find the right binding site, then they're stuck there until you degrade the DNA. But in the case of trabectedin, it wanders up and down the DNA and trying to find its thermodynamically stable sites. So next slide, please. So this is the reason that trabectedin is reversible. This actually shows the alkylation mechanism. So this is the reactive carbanolamine, the NCOH, and that undergoes loss of water to form this iminium species. And then N2 of guanine, which is in the minor groove, then alkylates this site on the drug molecule, and you form this covalent adduct. What is special about trabectedin is that because the groove is wider, a water molecule can get in there, and that actually facilitates the reverse reaction. So when the drug covalently bonds, it can reverse and go back to the free drug. So it wanders up and down the DNA, whereas the other drugs in the series, when they hit the right sequence, they stay there and don't move. So the person that did this work was Maha Zuwail, and she was actually the daughter of a Nobel laureate, Ahmed Zuwail, from Egypt, and he won the Nobel Prize in Physics, I think in about 2000 and I think it was 2000, and he was at Caltech. So I was very fortunate. I was invited to give a seminar in the chemistry department at Caltech. So I was able to talk about his Ahmed Zuwail's daughter's work at the meeting, and showed a slide of her, and she just had a baby at that point. So it's sort of a personal aspect of this. So next slide, please. Okay, so now we're going to talk about going from targeting duplex DNA to targeting this four-stranded quadruplex DNA. So I'm going to start out by talking about what G quadruplexes are on DNA. So next slide, please. So in 1989, and this was a difficult time in my own personal life, I actually got divorced in 1989, and I spent a lot of time at home, really struggling with the personal things rather than the professional things. And one of the things that I did is I wrote a couple of review articles, and I had the time to think about what I wanted to do for the rest of my career. And a guy called Alex Rich had just published the structure of a quadruplex, a four-stranded DNA structure, I think in Nature. And that intrigued me. And I started to look at the quadruplexes and think about them as a medicinal chemist, as an opportunity for drug discovery, because this was four-stranded rather than two-stranded DNA. So that's why I started to think about selectivity, and maybe you could get a better selectivity if you targeted four-stranded rather than two-stranded DNA. So on the next slide, this is a photograph of one of my wife's paintings. She's a watercolor artist. So next slide, please. So this is what a quadruplex looks like. It's the top right-hand structure, and this is the quadruplex that's found in the c-Myc promoter. So this is part of the control of gene expression, and in this case, c-Myc. So this is all mostly guanines. And so you see on the left-hand part of this slide, upper part, you see four guanines that are hydrogen bonded together. So these four guanines form this flat plate. And if you have three of them, they stack together and they're tied together by, for example, there's a adenine here, and I guess there is adenine, I think it's another base here. So they're tied together and it folds up into this four-stranded structure. On the opposite strand, it's obviously cytosines, and this forms a structure called an I motif. And these two structures can be formed from duplex DNA. So on the next slide is duplex DNA. So can you press the, okay, so RNA polymerase comes in, and then you get, with SP1, you get negative superlicity and you get unwinding of the duplex. And then that finally gives rise to a single-stranded region. And this is the guanine-rich, and this is the cytosine-rich. And so then you can form either a quadruplex, a G quadruplex on the G-rich strand, or a C quadruplex on the other strand, and press once more. And these two structures are mutually exclusive. You can either get the quadruplex or the I motif. So it was with this in mind, and also we knew, actually some years later, that these quadruplexes are concentrated in promoter regions. So if you look at the frequency, you find that quadruplexes are found in the promoter regions, and therefore they're probably involved. At least one of their functions is almost certainly, in fact, it is control of gene expression, or it's part of the system. So next slide, please. So in 2000, I'd been at Texas for 20 years at UT Austin, and I was recruited to Arizona in 2000 by this guy, Dan Van Hoff. And Dan I had known for about almost 20 years. He's a clinician, and he became head of the cancer center and he was head of the cancer center here for about 4 or 5 years. And then he moved up to Phoenix, and he's still at Phoenix at Tijan. So next slide. So Dan became a critical partner for me in my future translational research. And I first met him at an American Association of Cancer Research meeting in San Diego. And I was in Austin and he was in San Antonio. And I became director of chemistry at the University of Texas Health Science Center. And then when he moved from San Antonio, when he was recruited to Arizona, he then, alongside the dean of pharmacy, they recruited me to move to Arizona, and I was in the College of Pharmacy and in the chemistry department and in the cancer center. So that was my role when I moved here, primarily as a researcher. And then in 2001, I started my first company, which was called Cytonex, and we actually started in San Diego. Back in the early 2000s, there wasn't really a good tech transfer office at the University of Arizona, and there was actually not that much funding, so we had to do the company in California. And then Dan actually introduced me to PharmaMar in 1999, and then Race Oncology in 2025. And I serve as a consultant for both of these companies, as well as the company I founded back in the early 2000s. And translational research requires basic science, chemistry, and biology, critically clinical translation, and then a biotech entrepreneurial spirit. So those are the essential constituents. And I've started 3 different companies since I moved to Arizona. So next slide, please. Okay, so a critical paper that we published in 2002, which was in PNAS, we actually submitted this paper to 3 journals. We submitted to Science and Nature and one other journal, I think it was Cell, and they all got rejected at the, it wasn't even sent out for review. It was rejected by the editors, but finally we got it accepted in PNAS, and only because one of the reviewers, there was 3 reviewers, 2 were negative, and the third one was positive, who said, "I don't know whether this is true, but if it is, it's important." Well, it turned out that it was true. So this paper has now been cited 2,700 times. And what we showed in this paper is that when we took 2 cell lines, one called ramus, and there are cuts in the chromosome here and here, and then rejoining. And in this case, with ramus, you actually retain the quadruplex, whereas in the other cell line, CA46, the translocation is such that you lose the quadruplex. So we did a relatively simple experiment. We took 2 compounds, one that was quadruplex interactive, another similar one that couldn't interact with the quadruplex, and we looked to see if we could downregulate c-Myc, with the idea that if the one that interacted with quadruplex would stabilize the quadruplex, it would lower gene expression. And so in the ramus cell line, we found that the active compound knocked out MYC, whereas in the CA46, which lost the quadruplex, it didn't affect gene expression. So this is probably the most important paper from a basic science perspective that I published. And I used to tell my students, the more times you get rejected, then the greater the opportunity to get it highly cited. And that certainly happened for this one because it's my highest cited paper. Anyway, next slide. Okay, so I started Cytonex and actually while I was at Texas in the late 90s. And then because I moved to Arizona, we actually formed the company in Del Mar, just outside San Diego. So it was a new technology. We were targeting quadruplexes. I had a clinical partner, that was Dan Van Hoff. And then I used TLA, and we had patented the idea as well as a group of compounds that targeted quadruplexes. So we had the intellectual property. And then I found a CEO, a guy called Tom Farrell, that had a BS in engineering from Cambridge in the UK and an MBA from Stanford. So I became this chief scientific officer. He became the CEO, chief executive officer. And then we actually found investor support in San Diego. And we initially raised about $3 million to start the company in Del Mar. So next slide. So and this is sort of a bit off track from what I've been talking about before, but this was the initial drugs that we developed that were taken into the clinic. And we started out with compounds that Abbott had, and I'd worked with Abbott and were familiar with these compounds. And this was a duplex interactive compounds. And when we designed and synthesized molecules that were quadruplex interactive, and then we licensed these compounds to what was first Cytonex and then became Silene Pharmaceuticals. And from that, we got two drugs we took into the clinic. One was called Corfloxen, Quar because it was quadruplex interactive, and Floxin because it was a fluoroquinolone, and then CX5461. And this compound is actually still in the clinic. And in fact, it's now owned by a Taiwanese company. So next slide, please. So this is actually how the precursor QQ58 and the green spheres at the top show how the drug is interacting with this four-stranded quadruplex. So this is a model of the complex. Next slide, please. And this was the first patient that was treated in San Antonio. And like any medicinal chemist, I was very proud to be at the bedside of the first patient that got treated with a drug that came out of my lab. That was also a first-in-class drug. It was the first drug that targeted quadruplexes. But it wasn't this compound didn't make it past phase 1, mainly because of the pharmacokinetic problems. And that's why we went to another compound called CX5461. Okay, so that gives you an idea of what the translational process is. So now back to trabectedin. So as I told you, Maha Zawale had shown this interacted with duplex DNA. And what was different about trabectedin to all the other drugs that had this carbinolamine is this one was reversible on DNA, whereas the other compounds were irreversible. So in about, I guess about 3 or 4 years ago, because I knew a lot about quadruplexes by that point, I started to think, how could we explain the considerable increase in potency and selectivity? And I thought about quadruplexes. So next slide. And the reason I thought about quadruplexes is the clue, if you like, was this compound, ET743 or trabectedin, was very active against a childhood cancer, it's a bone cancer called Ewing sarcoma. It's a soft tissue sarcoma. And it turns out, and we knew this, that there's a protein called Ewing sarcoma protein that is really critical for targeting this cancer. But it wasn't understood how this protein could be involved in the mechanism of action. And I had seen a paper published in 2015 that said this protein actually interacted through this arginine glycine called RGG3 domain. It actually interacted with quadruplex structures. So that got me thinking that maybe the target was this protein that interacted with quadruplexes. And so we started to look for how this could be true. So on the next slide, we then found that some quadruplexes, including the one that interacts with Ewing sarcoma proteins, have loops. These are hairpin loops, duplexes, and this one has a AGC duplex sequence, which is the 3 base sequence that is targeted by trabectedin. So then what we did was to see whether the compound would be more stable when it bound to a loop associated with a quadruplex rather than just duplex DNA. Because remember, duplex DNA, the drug reverses and it wanders up and down DNA. And we wondered whether it would be more stable if it bound to a duplex associated with a quadruplex. So we used a variety of techniques, including mass spec as well as CD, and we showed indeed that if the drug interacted with a duplex associated with a quadruplex, it was stuck there. So what the drug would do, it would wander up and down the duplex DNA until it found a duplex sequence that was associated with a quadruplex, and that's where it would form an irreversible covalent adduct. So that explained the potency and also the selectivity. So this drug wanders up and down the DNA until it finds the right duplex sequence associated with a quadruplex. So that's how the drug has its selectivity. So next slide is the cellular and clinical activity of trabectedin and another drug that is actually a chemical related compound that PharmaMar synthesized. And so the next slide shows what happens when you add the drug to a cell and then use an antibody that is specific for quadruplexes. And what you see, as you increase the concentration of the drug, the number of fluorescent spots which are associated with quadruplexes increases. So this sort of proves at a cellular level that the drug interacts with quadruplexes and stabilizes them. And indeed, we now know what the quadruplex is that in the case of Ewing sarcoma, the quadruplex actually binds to this Ewing sarcoma protein, and that immobilizes the protein bound to it, and that has downstream effects. So that's how the drug works. And the other compound, lurbinectedin, also targets a different quadruplex, and this is actually involved in CPG islands, which is an epigenetic consequence. and the the lurbinectidine actually is used for small cell lung cancer and for melanoma. So you change the structure of the C subunit and it targets different quadruplexes and it targets different cancers. So next slide. So this is sort of illustration of how the selectivity. So if you just take saffromycin or anthromycin, this, when it finds a thermodynamically stable site on duplex DNA, it's irreversibly bound. So that's where it stays. However, if you have trabectedin, it sees the duplex sites, but it jumps out because it's reversible. And it's only when you get this thermodynamically stable site that it actually forms a irreversible covalent attachment. So that's the difference between anthromycin, saffromycin, and trabectedin. And this increases both the potency and efficacy of the drug. So this explains why we have the compound is so selective and also so potent. Next slide. I'm almost through, I think. So what lessons have I learned in this? I guess it's a 60-year journey, I guess. So next slide. So first of all, I think interfacing different disciplines. So I started out as a synthetic and biosynthesis chemist, and then got into molecular biology, and then of course we got into the clinical aspects, and then with people that had a business background, then got into starting companies. And, that's what it takes in order to, at least for me, to do something that was translational that I found very satisfying. The other thing, of course, is taking risks. And I only told you about the successes. There were lots of failures along the way. And that's what you have to do. I mean, you can't take risks without some failures. But then I also found that following my instincts rather than playing it safe, and simple things like finding the initial structure of anthramycin in the Journal of Pharmaceutical Sciences at the University of Maryland in 1971 was a lucky thing. It almost seems more than luck. And then lastly, and certainly not least, is finding the right people to work with. And I was very fortunate. I had an assistant, David Bishop, who actually had a PhD in music at the University of Texas, who worked for me as a work study student and then became an instructor in music at UT Austin, but preferred to work as a executive assistant with me. And so from 1983 up until, actually he still works for me. He left Arizona about a year ago, but he's still working for me. He's now in Cleveland. And I mean, without David, I wouldn't have been able to do a lot of what I've done. Dan Van Hoff has been critical. And then I've had some really talented graduate students and postdocs over the years. And this is really the team. I mean, to be perfectly honest, I don't think I couldn't have done this without these other people. And lots of the graduate students and postdocs, I think, were brighter than me. But I pulled them together and then interface different disciplines. And that's been the secret, I think, of the success I've had. So next slide. And this was my group in about 2014. We were in Bio 5 at that point. We moved from the Cancer Center, and I became associate director of Bio 5. in about 2006, and then moved into the College of Pharmacy. And this is David Bishop, who I just mentioned. David Bishop is next to me. I'm on the extreme left, or in this picture, I guess, on the extreme right in a blue shirt, and then David Bishop's in the pink shirt. And these are mostly graduate students and postdocs who've all gone on to primarily industry, but some people into academics. I have ex-students who are all over the place, some back in Europe, some in China, in Taiwan, and at universities in the US, but primarily probably pharmaceutical industry, places like Merck and Eli Lilly and so on. But it's been great. I've had a really good time. And the last slide, I think, is to go back to what really is equally important is one person's life. And this is me, I guess. It was 20 years ago when my eldest daughter, Bridget, got married. This is my wife, Terry, who's the watercolor artist. And then my youngest daughter, Nicole. Both of my daughters tried to avoid me and went into engineering as undergraduates, but then both ended up at Georgia Tech in bioengineering. And now Bridget is a VP for clinical sciences at a company out in Irvine, California. And my youngest daughter is in Dallas, and she works for a company that's making artificial devices, primarily hearts and so on. So I've been very lucky. I've been very lucky both professionally and personally. And I think my last slide is thank you very much. If you can go to the next slide. That's it. So thanks very much. And I went on longer than I wanted to, but if you have any questions, I'll be happy to try and answer them for you.
Yeah, thank you so much. That was really interesting. And I might reveal a little bit of ignorance when it comes to epigenetics, but you did touch on CPG islands, and especially thinking about how guanine rich and cytosine rich the quadruplexes are there known chemicals that can induce epigenetic changes in these promoter regions via the quadruplexes?
Yes, so I didn't go into the details, but both at the RNA and the DNA level. So at the DNA level, it's 6-methylguanine, and the enzyme that controls the methylation is a protein that binds quadruplexes. So if your drug binds the quadruplex, in the case of the second drug from PharmaMar, it actually prevents the protein from binding. It's called DNMT1, and that then it prevents the protein from controlling the methylation, and you get down regulation of gene expression as you get more methylation of the CPG islands. And then also at the RNA level, there's another drug that I work with from an Australian company that targets 6-methyladenine, and that again is a quadruplex binding protein. So quadruplexes are important in gene regulation, not only by directly binding proteins that are transcriptional factors, but also by targeting CPG islands and producing epigenetic effects. And all these things work through super enhancers. So it's the downstream events from the uh, from the promoter regions through super enhancers that actually result in the very potent effects on cancer on cancer cells.
That's really interesting. Thank you. I have another question that is completely different, but it's more on the human side of things. You were talking about how you found these amazing colleagues that you worked together as a team and you built this huge body of research and multiple businesses. And um, how do you identify the people who are going to be reliable and good teammates like that? I think
Yeah.
I certainly find it hard to evaluate people when I meet them and figure out what kind of character they have.
Yeah. Well, I haven't always been successful in doing this. So my first company, site that we called Sitenex, the initial CEO was a guy called Tom Farrell, who was very good. In fact, he's done extremely well since he left. But after I've been at Sitenex for about 6 years, we'd raised about 22 million by that time, and we were looking to raise a much larger amount. And the board of directors decided they needed a new CEO who had already raised that level of funds. So we brought in a new guy who I won't name, and he came in. I didn't like him, but I kept my mouth shut. I figured the board of directors knew far more about this than I did. And he came in and he said, I don't believe any of this quadruplex stuff. And he said, so we're going to forget quadruplexes. So the compounds that we already had that I'd shown were quadruplex interactive, he decided they were RNA Pol2 inhibitors, and they went off and into that area. I resigned in protest, and they spent almost $200 million trying to prove these were in the clinic, and assuming they were RNA Pol2 inhibitors. And in fact, in about 2015, a group at UBC in Vancouver showed that was completely wrong. They were quadruplex interactive. And at that time, a Taiwanese company bought the company, and I'm now working with them on these same compounds. So, uh, so in the business world, it's very different than academics, and my judge of people initially in the business world was not that good until I had some bad experiences, and then it's got better since that time. So, it's a trial and error thing. I've been very lucky. I mean, the CEO, the people I've interacted with primarily, like Dan Van Hoff and Tom Farrell and other people that I've worked with over the years, senior people, they've been very good. But there have been times when it hasn't worked out, and finding the right people, and also knowing when to say, "Hey, there's a problem here." I only had one case. I had unfortunately, a woman in my lab who actually created data rather than finding it. And fortunately, we never published anything with this data. But it was actually a graduate student at a research retreat who noticed that this student, all her error bars were the same. And he pointed this out to me, and that was the thing that tipped me off, that she had actually been fabricating her data. So, it's these journeys are not without their pitfalls. And that's been the same in my personal life too. You don't always make the right decisions, but then when you found you've made a bad decision, you need to do something about it, and that's the important thing. Experience is the only way that you're going to learn.
Well, thanks. I mean, I wish it wasn't only experience because that would speed up everything for me, but um it, gosh, that just seems like such a frustrating circumstance to be in with that CEO and such an expensive mistake for him to
Oh,
make.
yeah. Well, it's for the investors that put all their money in. They lost all their money. And I met with the board of directors um, after a meeting. I said, okay, I want to meet you uh, off site. And we met off site in Del Mar. And I told them, I said, either he goes or I go. And they said, well, we just spent $1 million bringing him from the East Coast to the West Coast. So we're not going to fire him. So as a result, they lost about $200 million because the target they went after was not the right target. And all the clinical trials failed. And I published a paper about, I guess in 2022, in which I described this whole thing, how we had started out with the right target, how someone came in and said it wasn't the right target, and then independently, a guy called Sam Aparico at UBC, who was trained at Cambridge and was an MD, PhD, and he showed independently that these indeed were quadruplex interactive compounds. But by that time, the company that I'd founded had obviously failed. And it was then that the Taiwanese company took up, and now they're back in clinical trials. And they actually have, I got an email yesterday, they're actually showing activity in pancreatic cancer, which would be incredible if that's true, because that's what my father died of. Anything else?
Oh, I just have a quick question for you. And this is coming from someone who has a very limited background in chemistry. What was it? Oh, okay. So I apologize if I misunderstood some of your work, but going back to the way that the trabectedin, is that how it's pronounced?
Yeah,
Trabectedin?
that's right. Yeah, that's correct.
Oh, yes. Going back to how it interacts with the quadruplex DNA structures, my question is is that molecule acting as an antagonist in a sense that it is binding to that quadruplex structure to inhibit the binding of other DNA binding proteins that could potentially promote oncogenic activity?
Yeah, there's nothing wrong with your question. That's a good question. So it goes both ways. In some cases, when the drug binds, it actually stabilizes the protein DNA complex. So in the case of with prostate cancer, we have a protein that I can't remember the name of it right now, that binds to the quadruplex. And when the drug binds to the quadruplex, it stabilizes the protein. In the case of the CPG islands, it actually prevents the protein from binding. So it can go both ways. And you're perturbing the structure of the quadruplex, not in the actual quadruplex, but actually uh, in the side chain. And if the protein interacts with the side chain or the loop, if you like, as well as the quadruplex, then that can actually prevent the protein from binding, as it does with Ewing sarcoma. But in other cases, it actually stabilizes it. But that's a critical point. What is the result of the drug interaction, and that of course affects the downstream effects.
Thank you.
Good question. Okay. Any other questions?
Yeah.
Oh, sorry, Jesse, you can go.
Yeah. I just had a kind of a specific question, but about the first clinical trial. Well, I don't know if it was the first one you ever did, but the first one you mentioned in your presentation,
Yeah.
where the drug got into phase one, but it didn't get beyond that because of the
Yeah.
pharmacokinetics issue. What were those issues? Like, what did you run into for that?
So what happened is we found that the drug bound very avidly to red blood cells, and red blood cells have a lifetime of about a week or so. So the drug was slowly released, and in fact, we tried to actually use that as a way to modulate the dosage in terms of frequency and so on. And that was a little bit successful. We worked with some people at Cambridge in the UK, but then we found that the CX5461 was much better. But that was the pharmacokinetic effect. It was extended the drug half-life. So instead of it being 24 to 48 hours, we could still find drug 1 to 2 weeks afterwards when the red blood cells were dying and releasing the drug. And that became a problem for us.
Did you try then modifying it? Maybe I missed that part, but
Yeah,
so that it wouldn't bind
that's
to red blood cells.
yeah. So that's exactly what we did. We modified the side chains once we knew that there was a lipophilicity problem. We then modified the structure of the side chains, and then we got rid of the red blood cell binding, and we made several hundred compounds. I'm just showing you. Well, I only showed you one, I think. But we made a lot of compounds over we started in about I guess in the early 2000s, and then after I left, after I resigned, they continued to make compounds in this series. I don't know how many they made in the end, but while I was at Cytonex, that became Silene, I think we made about 300 or 400 compounds.
Cool. Thank you.
Okay.
I have another question. I was thinking about how you got jobs. You were describing how you would in person meet with people and form relationships, and that day you get a job offer. Um, and one of the bizarre things about our time right now is that, of course, everything is online, and so anyone can apply. But because anyone can apply, there's so many people who apply that we've reverted back to needing those in-person relationships again to even be noticed and to get in the door. And so, as someone who was recently in a position of choosing which students would come to work for you or be postdocs for you, what would make someone stand out? What were you really looking for? And how did you think people most effectively approached you?
Well, first of all, I was never I was never just the one person to interview students. I mean, anyone that was serious about joining my research group, I used to have them meet with graduate students and postdocs. And of course, I couldn't always do that because the student was from Korea, then that would be a problem, but or a postdoc. I had to then rely more upon letters of recommendation and so on. But for graduate students, people would rotate in the lab anyway. And I used to get other people's opinion, particularly David Bishop. I mean, he had an uncanny ability to be able to pick out people that potentially would be problematic, although we didn't pick out the person who actually made up data in my lab. But I certainly didn't rely entirely on myself. I either used letters of recommendation, or when I could, it would be interaction with other people that I trusted in my lab. And, I had quite a few people that were senior people. For example, a guy called Vijay Gokhale, he joined my lab as a postdoc in 2003 or so, I think. He's now, I have a company that I currently I'm the CSO of, called Reglagene, and he's the head of chemistry. So he's been with me for 20 years. So I have some long-term people in my lab, and also my collaborators, of course, are long-term as well. So I guess the other thing that I've learned is experience. I mean, working in the business sector is very, very different than working in the academic setting. And I learned that the hard way. I mean, it's not surprising the objectives of people in the business world are quite different to the ones in academics, and they're quite different flavor. In fact, I don't know how true this is now, but most of the people I met in my early part of my translational career in the 2000s, they were Republicans rather than Democrats. In fact, the CEO of my first company, Tom Farrell, was certainly a Republican. He's not anymore, but he was back at that time. So it's different perspective, and it's just that was actually part of the reason that I started companies. I've been in academics from 1970 up until 1997 was the first time I started to start a company. So almost 30 years in academics. And, I've been through the cycle of writing grants, getting some of them funded, writing papers and so on, and having students. And I wanted to do something different. And I wanted badly to do translational work. And most of all, I wanted to fulfill what my father had told me I needed to do. And in order to do that, I realized I needed to get into the translational area. And, it's been actually a lot of fun, but it's also been tough at times. It's not all, as you'll find, I'm sure, or maybe already found, it's not always good. There are some bad things that happen. But, you ride them out, and then for me, I started to listen more to my instincts rather than just going by adding up all the pros and cons and then saying, okay, which is longer, the pros or the cons, and then making the decision. I've made some bad decisions using that as a way. It's much better if I listen to my intuition. I've become to rely on that far more in recent years. So I'll give you one example. I was trying to decide whether to move from Texas to Arizona. I had a very good setup in Texas, and I was treated very well there. And it wasn't an easy decision to move to Arizona. And one day, while I was trying to make up my mind, a colleague of mine walked into my office, a guy called Jim McGinnity, and he sat down with me and he said, "Listen, Laurence," he said, "you've got it made here. Why the hell would you want to leave?" And as he left my office, I thought to myself, that's exactly why I need to leave. I do have it made here. I want some new horizons. I want some new challenges. And that was the tipping point for me.
That's actually really funny. And this has nothing to do with cancer or biochemistry or anything. But I had this feeling on the first day that I went to a jujitsu class where I was so excited because I didn't know anything and there was so much to know. And it was just like this broadening of my world of, oh my gosh, there's so much to learn and I've got none of it. And I think that was a really surprising context for me to have that feeling because normally I have that associated with really just academics, but it feels great to be in that position and it feels a lot less compelling to me to be in a position where I know all there is to know about the thing.
Yeah. Yeah. I mean, climbing mountains, climbing up the hill is the fun part. When you get to the top, you can stop and watch and look at the view for a while, but then you need to find the next mountain to climb. Otherwise, it gets boring.
Yeah, absolutely. Does anyone else have any more questions? Awesome. Well, I really appreciate your time. This was very interesting. And with your permission, I would like to take the recording and share it with the other people in the group who weren't able to make it today so that everyone can watch it and see your very interesting journey.
Yeah, that's fine with me.
Awesome.
Thanks for the invite. I appreciate it. It was fun talking to you.
Thank you. Bye.
Bye-bye.